The next speaker is Professor Nirav Bhaat. Nirav Bhaat is an assistant professor at the Department of Biotech in IIT Madras, and he is also associated with several interdisciplinary centers, as you can see within IIT Madras. Above all, being a very good scientist, Nirav is a very close friend also, I think, a dear friend, I would say. So, a brief introduction. Nirav did his PhD at EPFL Lausanne. He has varied interests. Especially what I like about his work is that his work towards integrating in control theory aspects in biology. So Nirav, looking forward to your talk. Thanks for accepting our invitation, and over to you.
Yeah, thanks, Arijit. So as Arijit told me that I am actually started as a control theorist, and as you move on, I have been taught by many people computation and data analysis in some sense. So today I'm going to talk about some work we have started three and a half years ago on the understanding network biology, where I was trying to just write equations and prove some theorems. Some student came and said, can we reconstruct this some of the networks from the data that we used to do, but can we use literature data? And that's where we started our journey, and where I came to know something called large language model around 2018, where transformer models were built by many, by Google, and they have a first bidirectional transform BERT model. And we started with that, and now we see a lot of opportunities, and of course there are challenges. So today's talk is about, first I'll talk about motivation, why we need it. I think it will be very brief because all of you know about it as a network biologist, but still I will emphasize on that. And then we'll see that LLMs around us, thanks to ChatGPT this year and last year onwards, we have a lot of interest as a common man in large language models, and see what kind of attempts we have made to use large language models in network biology. And at the end, I will talk about certain opportunity and challenges and how we can actually work together to solve some certain problems. So biological systems, just wanted to give a disclaimer, I am not a biologist. I am learning most of the biology from my collaborators like Swagatika and the students. So biological systems can thrive, they can adapt to different environmental conditions, and they can fight diseases. And when it actually deregulated, we have a major problem because we have to actually make an intervention before the systems actually cease to exist. And that's where we are interested in. One of the reasons I started with biological system, because I like to see how system adapts as a control theorist. And then rest is the history. I started looking at fighting with disease because of collaborators. But what we do here is try to use network biology, network theory. So I use, in addition to network theory, control theory to understand biological processes as a network, such as protein-protein interactions, metabolic networks, gene regulatory networks, where I typically look at the metabolic reaction networks because of my past history with reaction networks. Now the idea here is use network theory to understand them, and people have been doing for last 2 decades. But one of the major advantages come from the omics data, which is high throughput data, where omics data has been used to actually construct different networks, understand what is the drug repurpose, for the drug repurposing, for metabolic engineering in processes, also designing the efficient experiments, looking at disease classification, biomarker discovery, and of course drug discovery, which is one of the holy grail in a pharmaceutical and biopharmaceutical industry. But typically people use this kind of data. When it comes to literature data, it is unstructured data compared to some of these data sets like fluxomics, metabolomics, proteomics, transcriptomics, and genomics. Other problem is that it is difficult to actually analyze unstructured data by the nature, and that's why we rely on the human experts. So typically, what happens that in addition to this, when people build some of these models, they also go to literature, curate it, the new reactions, understand what are the reactions exist in a certain particular context. And it is a time-consuming task. Some of us has actually done that, and we can actually vouch that when it comes to unstructured data analysis, it is not straightforward, and that's where the large language models have actually come into play. How we can use the literature data or unstructured data to perform some of the tasks, generate the hypothesis, build the model, is the question we have asked a couple of years ago. And that's where we actually ended up to large language models. So large language models, of course, ChatGPT, which is a poster boy right now for most of the, what you call it, generative pre-trained transformers. Google also has a model called PaLM, Pathway Language Models, which is even further, and the way I understood, they can actually not only take a literature or text data, but they can also take any kind of data as a token, like images and etc. Recently, of course, Facebook did not want to be behind, so there's a Llama, which has been there by Meta AI. And there are many other models available, like Bloom's, Galactica, Flan, T5. Even when I'm talking, there might be new model is getting released for certain tasks. And of course, when it comes to large language models, biologists are not behind. People have started looking at the biomedical counterparts as a BioBird, BioBigBird, BioGPT, etc. And one of the things, most of these particular modeling techniques, as a computer scientist, has a set of downstream tasks. They are interested in understanding if I can actually get an information extraction, okay, and in a certain manner. Can I do some sentimental analysis? Question answering is one of the major tasks. What you call computer scientists look at it. Image caption, object recognition with image plus text, and instruction following, text summarization, etc. And can we explore as a network biologist some of these tasks, or use this foundation model or large language model to create tasks for ourselves? That's the question we have asked. And with that, what we proposed is, let's start with the simple one: how to reconstruct the biological network from the help of the literature data. So what we want to do is, we want to actually go to the literature, let's say abstract, and use that abstract. So we here want to be context specific, so I don't want to be generic. So let's say I have take a context, let's say cancer, and in that cancer also take a specific example, let's say blood cancer or ovarian cancer. Then we'll collect data corresponding to ovarian cancer. That's very easy. Nowadays we can actually do it with a simple one Google, and we can even organize this in a very simple manner. And take this data, apply through large language model, and get a context-specific literature. And once we have context-specific literature, what we want to use LLM for is to build a network altogether. When I say network, what we are interested as a biologist or network biologist is different entities. You can see that there are three networks I'm talking about. One is the signaling networks, metabolic networks, and gene regulatory networks. Let me start with the metabolic networks. So what we are interested in is following. I want to know what are the metabolites present in a particular context. What are the reactions taking place in that particular context, and whether it is an irreversible reaction, reversible reaction. And I can go further and talk about metabolic reaction happening in different part of the cells and the transportation reactions. And this information, if I can actually get from these LLM models, then actually I can automatically build a rough model for particular context, which I can refine with more omics data, fluxomics data, etc., which are typically targeted towards that particular diseases. Okay. And I was mostly interested in finding out simple relationships, and one of my students, Pawan, said that can we actually do who was working on context-specific models, can we do actually for the context-specific. And there they started with something called COSMEL, which actually is a transfer-based approach to build the context-specific metabolic models. So what we do in typically context-specific metabolic models currently is we use context-specific transcriptomics data. So let's say I'm interested in understanding a particular liver cell, brain cell, or even cancer cell line. That's a particular context I'm interested in. Then I'll get a RNA-seq data or transcriptomics data for that particular context. where gene versus expression level in a different context, and use some thresholding method which allows me to provide certain co-reactions and use a super reaction structure, or what we call is super structure of reactions, which is currently is Recon 3 or Recon 2 to actually use along with this particular thresholding method and model extraction method to get for different context different kind of models. And then I can use this particular model for doing whatever downstream task in terms of simulations, whether I want to understand what are the reactions taking place, what is the flux through it, if I perturb it, what kind of reactions change in the reaction, etc. So this is a downstream task afterward, which is mostly simulation studies. But building these models is cumbersome. Often you require not only data, but also you have to actually manually curate through the literature and find out what are new reactions are taking place. So what we try to say that instead of using the thresholding methods, can we actually use literature data and transfer-based model to get the context specific reactions or what we call co-reactions, and then we can use Recon 2.2 to actually get our context specific models. Of course, there is a interesting fact that over a time we realize that we can actually update the Recon also. So this is what we are trying to do, is trying to see if we can find some new reaction which is not there, and we update that, which will help us to actually expand the human metabolic reconstructed models for the reaction 2.2. But right now we are looking only for co-reaction for particular context using the particular context literature data. So how we do this? So first we have to actually extract the entities. The entities like enzyme, reaction, gene. So we took a context specific literature data. There is again there is a LLM model, which I'm not going to discuss about it, which actually retrieve automatically the important abstracts for particular context like cancer, ovarian cancer I'm interested in. So currently human is doing it. They are collecting all the abstract for the ovarian cancer, and typically these are PubMed abstract. And we use NER task, one of the large language model, to figure it out what are the enzyme, gene, metabolites and other entities. Once I have these entities, I'm also interested in the relationship between them. Once I have these entities, we use GPR rule to actually, and Recon 2.2 to actually construct the or predict the reactions which are actually happening in this particular context. And then with the transcriptomics data and the model extraction method, we actually build the models. So this is a simple work we started, where we used the available BERT model in the literature, and we realized that it does not give that good results, because this model is actually trained for generic tasks. Basically, it's trained with the Wikipedia articles and those kind of articles, but not for a biomedical articles. So another problem with this one is that it only uses the abstract, which is very small text instead of the large complete full text. And that's where we started to see if we can move from the abstract alone to the full text information. And often in the science, it is not, we have like to write big sentences, compound and complex sentences in English. And in that cases, some of the current models cannot actually handle the text, because it has at a time it can has a certain tokens, or you can think of words it can actually process. And the question we are asking, can we actually do full text, and the larger paragraph can we actually process in one go? And there was, of course, not only we, only computer science community was thinking, and they have something called BigBird model, which actually can take a full, not full text, but larger text compared to the current models. And what we did is we actually used PubMed abstract, full articles, and mimic 3 data sets. So mimic 3 data sets is a data sets talk about 40,000 patients related what you call documents, their electronic data, most of the text, what doctor described over the years, and we use that data together to actually build a BigBird model for biological tasks. And the whole idea here is to can we use this for getting a better name entity recognition for biological context, relationship extraction for biological context. And typically when you build these models, you have to try with the benchmark data sets, which is called BLURB. So we actually, what we did is we tested our model, which we have trained, with some of the recent model available in biomedical domain, which is PubMed-BERT, BioLink-BERT, which is recently released. There is a large one. I'll talk about what does it mean between base and large. And BioBigBird, which we have trained in a different manner than the typically people do it. So we started with a simple PubMed abstract, which we call stage 1, to understand what is the impact of the full text when we actually do it. Then we actually train the same model again with the full text. Of course, this is full text. We removed headers, some of the non-useful informations, and only took the informations which is useful. And then Mimic 3 data we have actually trained in the third stage. So there are BigBird model. We have 3 kind of models which are trained on abstract, then full articles, and on the Mimic 3 data sets. And this, just to add, this is sequential addition of each data set. Okay. Now, this BLURB is actually it's a data set which is called benchmark data set, where every model actually try out how good they are in terms of their model. And here there are different tasks. For example, the first couple of tasks are name entity tasks. It means we want to figure it out what are the name entities in a given paragraph. As a human being, we can actually do it very well. Okay, if you are not biologist, you would do little poorer than on the biology text. But can model do that well? And we saw that in many tasks, we have decent results. Of course, we are compared with the large model, which has a 340 million parameters, compared to our model has a 130 million parameters, which is, so our fair comparison is with them. But we were excited on some of the tasks with the lesser number of parameters, we are doing far better modeling. And compared to ChatGPT, we are almost 2,000 less number of parameters, 2,000 times less number of parameters. And compared to Palm, we are almost 5,000 less number of parameters. Just to show that with a smaller model, we can do some tasks far better. Then this CAM plot is about relationship extraction. Here is a gene and disease network. Here is a chemical and protein networks. Here it is disease network. And you can see that by using the more text or the full text, we can get a far better dependency compared to some of the base model, or even compared to the larger model. PubMed question answer, and these are question answer data sets where the large BioLink-BERT performs better, where we perform marginally poor, and we are trying to see why it's happening right now. On the HOC, which is a documented summarization task, where we are doing extremely well compared to any other task. So it tells you that some of the tasks, when we actually have a full text, we get a better and better models, and that's what we got it here. And we are saying that we are waiting for the reviewers' comment about other tasks. So here, if you look at it, all this bold one is where the model is performing better. But here, remember that here the number of parameters are almost 3 times more than what we have, which has its own impact on the models. And we are trying to see if we can improve it by further training. Unfortunately, for that we require a lot of large computational power, and we have been in a process of getting a grant from the Google to see if we can actually do that. Now, about me, of course, only not our group. There are other people who are doing different tasks. In our groups also, we actually use LLM models for drug discovery. We assume that all the molecules are language like SMILE and do that, but I'm not going to talk about. I'll talk about what other tasks people have done in the literature. One of the interesting tasks people have looked at the drug target interactions, predictions. where they actually take a drug target interaction pairs and drugs and the targets, typically targets are proteins, so you have a sequence of amino acids. Drugs are typically small molecules, so you have a representation of SMILE, and these both are basically a character, if you can think of, is as a sentence, because amino acid sequence is also some numbers, 20, what you call it, letters arranged in certain particular manners. Similarly, SMILE is also chemical structures. It has its own language, and it is also expressed as a sentence. So we think people have thought it as a sentence, and can we actually use this sentence to actually train large language model to predict the interactions. So these interaction pairs are given to them, and they used the BERT model here and got pairing for drug embedding and the protein embedding. Once you have this embedding, you can actually do drug target interaction for particular context. So remember that one of the main advantage here is that often we have a far larger data set for drug. There are, if you go to PubChem, there are around 107 million SMILE strings are available for different chemicals. Similarly, for different proteins, there are databases where there are a lot of protein sequences available. What we can do is we can actually get this embedding by using language model separately, and then use for our task, which is particular drug target interactions, where we have a very small number of data sets, use this embedding to actually predict, depending on the task, whether it's a regression or classification. Typically it's a classification, whether we want to know that new drug has what kind of targets, or can it work on specific targets. So this is another one applications people have looked at it for drug target discovery. Another one people have looked at is protein-protein interaction predictions. Again, as I told you, protein sequence is taken as an input, and they use what they call masked language modeling. In other words, they mask part of it, part of the sequence. It means these are the BERT model does not know what are those sequences are there. They use rest of the sequences and tend to predict what will be the sequences. It's like a filling the blanks kind of approach they do it, and that provide you again better results. And then for a particular task, you can actually fine tune it and predict the protein-protein interactions. So there are more and some more like for metabolic networks, gene regulatory networks. There are some literatures, but not as many as we want as a network biologist. So people have started to work on it, and at the time what we were discussing, that if I want to do a particular task, are the data will available? Because if I have this database for the protein sequences, drug, then gene, but I don't have a data set for my context. And there were a lot of opportunities are available to generate the data and apply these models. Of course, there are challenges. As I told you, you have to have a good quality data. So LLM for network biology task, so typically this is what we define. There might be more tasks, and we are happy to learn what are more tasks you have. One of the tasks which we look at is getting the context-specific abstract itself. How to have a information retrieval automatically, which provides you a good context-specific literature. Currently, if you look at the Google Scholar, it provides you, correct? It's not denying, but it does not provide that good information. If you have to search for it. So we are trying to see, can we actually create a context-specific information retrieval which is targeted towards particular task. There is a work we have done, very interesting work by one of our dual degree student, but I'm not going to discuss much. But yes, this is one important task, and for that there are not good data, because if I have to go to the literature and get a paragraph, how good it is for my task is not known. So you have to actually have a manual annotation that this is a good example. Now, unfortunately, most of the time we have good examples. There are not negative examples, because people ignore negative example and have a positive example. Correct? How often, if particular paper is not useful, we do not remember it. So creating a negative example is important for automatic retrieval. So we have come up with some pipeline where we actually generate automatically positive example and negative example. But for particular task, we do not have a data. So right now we are using the benchmark data sets where we are doing well. But generating data set for this itself is a one task in hand. And then use that for using even not what I'm training, but any retrieval available for you. You can use it with your task and get a good model. Another task we are looking at relationship extraction task and name entity recognition task. Typical NER tasks are mostly for enzyme, protein, gene, and some molecules. But biology has many molecules, different kind of RNAs and different kind of molecules. So can we actually create data set for that is another question we are asking. Relationship extraction task, typical again, relationship is in a biology not only about one molecule to another molecule, but what kind of relationship is it activation or inhibition. Sometimes different molecules have a different effect on each other. For example, protein and gene might have some interactions, or some metabolite has some effect on gene. So those kind of tasks is also we are interested in, where not only relationship but directed relationships also. So these are some of the interesting tasks has to be actually built to actually data has to be built for this. So for the name entity task, we have actually protein functional classification is still a problem. So people have not done that. So we actually get the data for that, or even EC number classification, which is again kind of interesting topic. People are using sequence to predict what is the EC numbers. Transcription factor, whether they're activator or repressors, those kind of data are not available. Again, organelle identification. This is another interesting question asked by one of the colleagues. Can you actually get what experimental techniques are used for a particular context? And can we actually predict it? So if I have some question, I want to do it, I can go back and use this to quickly figure it out what kind of experiment techniques I should use, or even protocols. Because often optimizing the experimental protocol for particular context is very difficult. Drug mechanism action is again, there are no data available. Relationship extraction, this is what we are interested in, but there might be more on that. And we started some work with a couple of colleagues, students, who are trying to actually, who have set up at least within the IIT a framework where if I give you abstract, human can actually annotate manually different kind of entities, relationships. They can define new entities, which is not there in our this one. And we are hoping that we can actually make it public and take your help to actually annotate and make it as what you call it, community efforts to get better and better data for ourselves and use that for getting a better and better models and use for whatever task we are interested in. So let me conclude. Large language models brings a lot of opportunity for network biologists. We have not done much work yet, and it's and of course for the biologist. Domain specific training for LLM is the key for building a models. You can't go to ChatGPT and say, answer my biology question. In fact, ChatGPT cannot even answer a simple question which is supposed to answer because of phenomena of hallucination. So, and now people are unhappy with the ChatGPT. As you go, you have more data and you realize that performance is poor. So domain specific training is a key here. One of the challenge for the network biologists is the scarce data set, because we do a lot of tasks and we do have to do manually curation. Can we actually do it without manual curation? So hopefully maybe by the end of this month we can actually give you publicly access. So generate the data sets, and if you let us do as a community and generate the data. I would like to thank Google TPU providing a good amount of data, computation for doing some of this training, and also RBC Design and IBSC for computational support. Pawan, Roshan and Rajiv are the one who actually come up with some of the good ideas. which have been implemented by some of these dual degree students along with us. So if you are interested in developing data sets, please write to me, and we'll actually share with you the data we have. We have a huge amount of what you call PubMed articles processed well, and you can actually use that for your research. Thank you very much. And if you have any questions, I will answer.
Thanks, Nirav. This is really exciting. So we have time for a few questions. So questions?
Hi Nirav, nice talk. I'm Rahul Sadathan from here. I don't think we met, but I was going to ask a question about hallucinations, and then you said something in the last slide. So my understanding of hallucinations, basically these language models don't have any kind of semantic comprehension. They are a probability distribution over strings, basically. That's, except they're very sophisticated. So an example of a hallucination is you ask it what literature reference supports your claim that a certain gene is regulated by a certain factor. It'll actually make up something that looks very realistic because it is just learning from what citations look like, but the citation doesn't actually exist. And similarly, you ask a language model about reaction pathways and so on. It can just make up something plausible which has no basis in any of the papers. It is putting things together in a way that syntactically makes sense. How do you get around that, even if it is a domain-specific LLM?
So at the end of the day, you have to see how we can, one way we can do it by bringing the more science to it. For example, reaction. So we look at the thermodynamic feasibility of the reaction by looking at Gibbs free energy or those kind of aspects. Then there are other checks, domain-specific checks, which we have to do it before we go and do the analysis. So that is a one way to actually be ensure that whatever we are testing is correct or not. Other way, I think all the new models, data-driven models has a robustness problem. Not only, let's not make it alone LLM models. Even simple neural network models also has a issue with. So we have there's a huge work in the robust training of data-driven models. So at the end, what I can say is that you have to actually bring the science field to it to actually screen the data. In our case, we use some of the domain-specific tests, like as I told you, reaction. For reaction, we look at conservation of atoms, then look at the delta G of reactions to find out whether this feasibility or not is there or not. Otherwise, it's very difficult to tell. So in other domains, you have to bring your domain-specific knowledge to make sure that whatever answer you got has a meaning. And that's what people realize, that they are not getting the answer. Simple, for example, itinerary. I know many people who tried ChatGPT for itinerary. Like they want to go to new town and want to see what, so they put their own town and see that some of the places are mislabeled. So that way you can come to know that it is not reliable. And now then you can correct it, and of course, hopefully ChatGPT is learning from that by some feedback and improving it. Otherwise, I do not see any other way we can rely. Means I won't take every time output we'll take up with pinch of salt. I won't take as a absolute truth.
Rahul, any follow-up? Okay.
Yes, so wonderful talk, Nirav. One recommendation is that in your PubMed, like PubChem data, a single smile, you're using that as a string, right?
Yeah.
So for representation of a single chemical entity, there can be up to 10 to 12 smiles. So instead of using smiles, if you use the fingerprints, it might give you a more precise understanding of what you're predicting in your DTIs.
Okay, so apart from smile, we are also using sometimes. We are not. This is not my work. We are using smile. Okay, that's true. Also, we are using functional group. That is one of the things to ensure that. A third thing we are using, because there is a single line, so we also use some other information for 3D stereo symmetry and those kind of things. But yeah, that's a valid point. But functional group, we have for property prediction, we have outstanding results. So we are writing it up, and we realize that you don't need any other information except the functional group information.
Questions?
Hi, I'm Ravi. So are these publicly available, the access, BioBigBird?
currently, no, we will make it once the publication is out.
Okay, great.
So where do you take the training data sets from for different domain models?
Okay, that's what is the challenge. Okay. So we have some training data set for some of the purposes we have. Currently, we are not doing that. So for this one is benchmark data set. Okay. And the remaining model which has been trained, we actually manually means we scrapped it by ourselves. These PubMed articles anyway available. PubMed abstracts are available. PubMed articles, one student has put a lot of efforts to scrap it, clean it up, and before with some manual checking, time to time random checking that we are getting the proper things. Mimics 3 is already curated, well curated. We got it from the lab. So all this data, okay, other data also which I want to tell this has been curated by different people manually, and that's what is one of the challenge for network biologists, because there are a lot of other tasks we do not have. For example, your problem, there is no, you have to do manual curation. So can we actually get the data with small data set which allows us to curate a larger data set, unlabeled data with the small labeled data? Can we do it is the question we try to answer, and for that we at least have to have small unlabeled data sets.
So we thank Nirav for this wonderful talk.
Yeah, thanks.
Thanks, Nirav.